Hi, good morning. My name is Huabing Wang. I'm a pathologist at MD Anderson Cancer Center. I do a lot of collaborative research on the pancreas cancer, and also I have a lab running for the to examine the mechanism in pancreas cancer, trying to identify molecular markers and the target therapies. So within the next 1 hour and a half, we will have 5 distinguished speakers. They will present their work from different angles to dissect the novel mechanism of pancreatic ductal adenocarcinoma and their application for the treatment. Before we start the talk, I want to tell you that please hold your question to the end of the section. So each speaker will have 15 minutes. So before we start the section, I would like to introduce my co-host, Dr. Nancy Du. She is an associate professor from Weill Cornell University. So go ahead.
Okay, welcome to the final day of the APA meeting. It's very great meeting, and my lab work on pancreatic mouse model of metastasis. It's a great honor to be here with all of you today. And I would like to introduce our first speaker, Christine Chu. She's an assistant professor at Columbia University. She has been accomplished a lot for the field, and today we would like to hear her research about methionine reactivity regulates pancreatic cancer metastasis. Welcome.
All right, good morning, everyone. I'd like to start off by thanking the organizers for this opportunity for me to share with you some of our recent work in studying reactive methionine and the process of pancreatic cancer metastasis. Okay, so a main question that we ask in my laboratory is, what does ROS do? This is obviously an important and also an old question, not just for pancreatic cancer, but also for pancreatitis. And I think a lot of you would agree with me that the role of reactive oxygen species, or our focus in the field on reactive oxygen species, has largely been in the process of damage. And this is, of course, really important because indiscriminate damage by highly reactive raw species, such as hydroxyradicals, would incur damage on macromolecules such as DNA and also lipids. But I think a lot of you would also agree with me that unless there is a very high level of ROS, most of the time, it's local bursts of ROS that happens within the cell, and that does not cause DNA damage or lipid damage. Instead, what happens is this ROS focuses on sulfur-containing amino acids. Those are the main effector proteins of local bursts of ROS. And the consequence of changes in ROS to sulfur-containing amino acids is that this leads to oxidative modification on free thiols, as well as other sulfur-containing amino acids that I'm going to talk to you about later on. And what happens is this brings about a structural and or functional modification of that recipient protein in a process that we call redox signaling. The best way of understanding redox signaling is taking the analogy from phosphatase and kinase-based signaling. But instead of asking the question, what happens to the structure and function of the protein upon addition and removal of a phosphate group, we ask what happens to the activity and function of the protein upon addition and removal of an oxidation mark. So most of our lab's previous work has focused on studying this process in the context of the amino acid cysteine, a very well-characterized sulfur-containing amino acid. But what I would like to share with you today is some of our more recent work in characterizing the role of this process in methionine, the other sulfur-containing amino acid. And so just like cysteine, methionine can toggle between its reduced and oxidized state. And here we have a very clear writer and eraser system. The writer, obviously, it's reactive oxygen species, mostly in the form of hydrogen peroxide, whereas the eraser, it's a methionine sulfoxide reductase called MSRA. And given our earlier work on cysteine, where we showed that oxidative modification of cysteines in pancreatic cancer cells can regulate transcript-specific translation, we ask whether a similar ROS signaling mechanism also exists through methionine. And so the first piece of evidence that hinted to us that this may be the case for pancreatic cancer came from a tissue microarray that we built together with our clinical collaborators at Columbia, as well as Tony Hollingsworth, who runs the Rapid Autopsy Program at Nebraska. And here we saw in a panel of patient-derived normal, pre-malignant, malignant ductal adenocarcinoma, and importantly, patient-matched liver metastasis, we saw that there was a very striking stage-dependent decrease in expression of this methionine sulfoxide eraser protein, MSRA. And that in the metastatic setting, the expression of this protein is almost completely undetectable. We can also model this in organoids. And here I'm showing you a panel of murine normal, tumor, and mouse match metastasis. And again, you can see a very similar expression pattern where going from normal to tumor to met, there is a stage-dependent decreased loss of MSRA. And this is good news to us because one, it validates the expression pattern in our TMA. And second, it tells us that we now have a model system to study what this means. This is important because a decrease in expression level doesn't always mean that it's functionally relevant. And so what we decided to do was we took tumor organoids that still express MSRA and we used CRISPR to knock out MSRA, and we asked what happens to primary tumor growth in vivo. And so like everyone else, we did the obvious experiment. We performed orthotopic transplantation of tumor organoids that are proficient or deficient of MSRA. And we asked what happens to primary tumor burden 5 weeks after transplant. The result was a little bit disappointing initially because we saw that comparing the two groups, there was no statistically significant difference in primary tumor burden. However, what was very striking was that loss of MSRA very dramatically increased the incidence of metastasis. Now, of course, you would sit there wondering, well, maybe this increase in metastasis, it's because there is a small, even though not statistically significant, increase in primary tumor burden. And we thought the same thing. And so we decided to test this a little differently by taking the primary tumor out of the picture and ask in a metastatic model, can we see an increase in metastasis? So to this end, we turn to the liver metastasis model, given that liver metastasis, it's the most frequently found metastatic lesion in pancreatic cancer. And the way we do this was we injected the exact same organoids that are wild type or knockout for MSRA, but this time through the spleen. And we assay what is the ability of these cells to migrate through the portal vein and to seed in the liver. And in this experiment, the result was crystal clear that loss of MSRA very dramatically increased the incidence of liver metastasis. Now, what happens if we do the opposite experiment, meaning instead of knocking out MSRA, what if we try to restore MSRA expression? So we generated metastatic tumor organoids where we introduce a doxycycline-inducible construct to re-express MSRA. And in this case, we again injected orthotopically these organoids into the pancreas. And here we waited for tumors to form, which we can monitor by ultrasound imaging. Upon formation of an established tumor, we then enrolled mice into either a water treatment or a dox treatment group. And we asked 3 weeks later, what is the difference in tumor growth? And the answer was exogenous expression of MSRA in metastatic organoids does not change the rate of primary tumor growth, which resonates with what we saw in the knockout experiment. However, if again, we turn to our liver metastasis model, this is when things get interesting because here we saw that re-expression of MSR significantly suppresses the formation of liver metastatic lesions. And most strikingly in this experiment, we found that every single mouse in this control group gave rise to extra liver metastatic lesions, whereas none of the mice in the dox group gave rise to extra liver lesions. So collectively, these in vivo experiment establishes MSRA, this methionine sulfoxide eraser protein, as a potent suppressor of pancreatic cancer metastasis. Now, what about metastasis does MSRA regulate? We know metastasis involves survival through the bloodstream, migration, invasion, a large number of processes. But we think that MSRA, at least in part, regulates the process of migration and invasion. And this came from this observation here. What we noticed was when we grow organoids that are normal ductal organoids or tumor that express MSRA in low concentrations of matrigel, they would still maintain their three-dimensional structure. This, however, was not true for those tumor organoids that are deficient of MSRA. We found that they would migrate through the matrigel and form 2D monolayer outgrowths. So this was the very first hint to us that MSRA may be regulating the process of cell invasion and migration. And so we decided to test this using a conventional wound healing assay. And what we did was convert 3 independent tumor organoid lines into conventional 2D monolayer cell lines. And then in a scratch wound healing assay, we asked what is the rate of wound closure. And after 36 hours, we found that the size of the wound in MSRA knockout pancreatic cancer cells represented by blue, it's significantly smaller than that of their wild type counterpart, indicating that the rate of wound closure when cells lose MSRA, it's much faster than the wild type counterpart. And the opposite is true when we try to express high levels of MSRA in these cells. Now we slow down the rate of wound closure. So what is going on? We want to identify proteins that are selectively targeted by MSRA that underlies this migratory and metastatic phenotype that I described. And the approach that we took was chemoproteomics. So conventionally, if you're familiar with cysteine reactivity proteomics, the approach that one would take, it's an acid-based approach using electrophiles like iodoacetamide and maleimide, which would alkylate free thiols in the cysteines. This gives rise to a formation of a derivative which we can measure quantitatively by mass spectrometry. This method, unfortunately, would not be suitable for methionine because methionine is a much weaker nucleophile than cysteine. And about 4 years ago, a paper came out from a chemist's lab, Chris Chang at UC Berkeley, reporting oxaziridine, an N-transfer oxidant to be able to selectively derivatize reactive methionines. And so we quickly reach out to Chris and ask if we can get some of his compounds to use to measure reactivity changes in the methionine proteome. And he graciously came on board this project. And so we took oxaziridine, which contains an alkyne handle that allows us to use click chemistry to add on a biotin tag for streptavidin enrichment. We used this probe to characterize our full panel of normal, tumor, and metastasis organoids. And the question in this experiment is, what are the proteins that undergo differences in methionine oxidation throughout the progression of normal, tumor, and metastasis? And importantly, in this experiment, we characterize not only the reactive methionine proteome, but also the total proteome in order to adjust for protein expression differences. Here's what the results look like. As a reminder, the way this probe works is it reacts with free methionine, giving rise to a derivative that we can measure by mass spectrometry. So loss of reactivity, it's an indication of an increase in oxidation. And I hope you can appreciate that going from normal to tumor to metastasis, there's a very clear stage-dependent decrease in reactivity of methionine-containing peptides, indicating that there's an increase in oxidation. And importantly, this decrease in reactivity very nicely mirrors the expression pattern of MSRA that I described at the beginning of my talk. So there are over 400 methionine-containing peptides that exhibit a loss of reactivity in the metastatic setting. How do we go about establishing function? We decided to take a reductionist approach, and we asked if we rank these peptides based on strength of oxidation, what do we see? And this was the result. We found that in the metastatic setting, the most strongly oxidized peptide maps to a protein called pyruvate kinase PKM. PKM perhaps does not need much introduction. It is a very important glycolytic enzyme that catalyzes the conversion of phosphoenolpyruvate to pyruvate. To determine whether this oxidation event on PKM is at all functionally meaningful, we did Seq13 glucose tracing to ask what happens to glucose flux in cells that are proficient versus deficient of MSRA. While we see no differential labeling in all of the proximal metabolites in the glycolytic pathway, we saw a striking twofold increase at the step of pyruvate. What this means is going from phosphoenolpyruvate to pyruvate, there's an increase in enzymatic activity. And indeed, we saw that all downstream metabolites in the TCA cycle also exhibit an increase in labeling. To establish that pyruvate kinase does exhibit an increase in enzymatic activity upon loss of MSRA, we immuno-enriched PKM1 and PKM2, the two different isoforms of PKM, and measured its enzymatic activity in MSRA proficient versus deficient tumor organoids. And as you can see, MSRA expression does not regulate PKM1, but it regulates the enzymatic activity of PKM2. In our mass spec experiment, we were able to identify 6 methionine-containing peptides that are reactive. But comparing the reactivity in tumor versus metastasis organoids, we found that only one single residue, methionine 239, exhibits a loss of reactivity, meaning it's oxidized in the metastatic setting. And this is where methionine 239 it's located. As you can see, it's solvent accessible, and it's very close to the FBP binding pocket of PKM, which is an allosteric regulator. And we'll come back to this later on. This methionine residue is highly conserved in evolution, and so this gives us additional confidence that this is likely an important residue. But to establish the functional significance of methionine 239 in regulating PKM2 activity, we mutated this residue to leucine. And we chose leucine because it is structurally similar to methionine, but it's not redox sensitive. And again, we measured the enzymatic activity of PKM2 comparing control versus MSRA knockout cells. And here you can see that consistently in 2 independent pairs of tumor organoids, loss of MSRA increases the enzymatic activity of wild type PKM2, but not the mutant. This establishes methionine 239 as the effector site of MSRA oxidation that mediates an increase in enzymatic activity of PKM2. So how does one oxidative modification change the activity of PKM2? I mentioned earlier that the location of this methionine residue, it's very close to the FBP binding pocket of this protein. And what this means is that the FBP binding pocket, it's an allosteric site that regulates the activity of PKM2, whereby low activity PKM2 exists in a dimeric state, whereas high activity PKM2, it's in a tetrameric state. And so we asked whether the oxidation state of methionine 239 controls the tetrameric state of PKM2. And we tested this using a non-reducing gel analysis to quantify tetrameric versus dimeric species. In the wild type protein, loss of MSRA always increases the tetrameric species. And this effect is completely abolished in the mutant, where methionine 239, it's replaced with leucine. The effect is seen more clearly when we do size exclusion chromatography to separate monomer from high molecular weight tetrameric species. Again, loss of MSRA shifts PKM2 towards the high molecular weight species, but this is only true in the wild type protein, but not in the mutant protein. And so finally, all of this data suggests that loss of MSRA oxidizes methionine 239 and PKM2 increases its enzymatic activity. But the key question is whether this underlies the migratory and metastatic phenotype that we see in MSRA knockout cells. And so finally, in this last slide, I would like to share with you this experiment where we used a small molecule called TEPP46 to increase tetramerization and activity of PKM2. In a wound healing assay, we show that activation of PKM indeed increases cell migration in wild type cells, and this phenocopies cells that loses MSRA expression. And importantly, MSRA knockout cells are insensitive to this small molecule inhibitor. And finally, in an in vivo setting using our liver metastatic model, we found that animals treated with the PKM2 activator significantly increases the incidence of liver metastasis. And so I'd like to close. Summarizing what I've shown you today is that loss of MSRA, a methionine sulfoxide eraser protein, it's a potent suppressor of pancreatic cancer metastasis. Using chemoproteomics, we were able to identify the peptides that selectively undergo oxidation. And for the interest of time, I only showed you one example today where we biochemically characterized PKM2 to be one such hit. And I showed you today that oxidation of the single methionine residue contributes to the activity of PKM2 by promoting its tetrameric state. And using an orthogonal approach, we established that activation of PKM2 indeed promotes migration and metastasis of pancreatic cancer. And with this, I would like to close and acknowledge all the people that did the work. Much of the work that I showed today was spearheaded by Hujin Feng from my lab with a lot of help from Justin and Aster. And this is a really productive collaboration with our chemist collaborator, Chris Chang, and his postdoc, Dan He at UC Berkeley. And of course, thank you all for your attention. I'm sorry I went over time. I'll be happy to answer questions later on.
Yes, thank you. Thank you very much, Dr. Chu, for the excellent talk. The next speaker will be a virtual presentation given by Dr. Hui Zhang. She's a professor of pathology at Johns Hopkins University. She's also the co-director for the mass spectrometry core facility. So her talk, it will be proteogenomics of pancreatic ductal adenocarcinoma. Please bring her presentation up.
I would like to thank the meeting organizers for the opportunity to present our work on protein genomics of PDAC. Pancreatic cancer is one of the deadliest cancer. PDAC represent over 90% of all pancreatic cancer and is mainly responsible for the majority of pancreatic cancer-related deaths. Numerous efforts to characterize genomic and transcriptomic feature of PDAC enable us to better understand PDAC. But the impact of these changes in PDAC are largely unknown. Technically, PDAC tumor is has a lower cellularity, which pose unique challenges for protein genomic characterization. This is the four topics I'm going to present today. First, we would like to address the lower neoplastic cellularity. Tumor tissues are isolated by bulk or by coring or laser microdissection, analyzed by histology, proteomics, and genomics. The data showing that the coring is not effective to enrich the tumor cellularity with the tumor contain lower tumor cells. LMD can be used to enrich tumors in all tumors, but enrichment effect is varied. The protein genomic analysis of PDAC cohort comprised of 140 tumor samples, 67 normal adjacent tissues, and 9 normal ductal tissues. A set of omics data, including whole exome sequencing, whole genome sequencing, RNA-seq, microRNA-seq, DNA methylation, proteomics, photoproteomics, and glycoproteomics are generated. Proteomics, the photoproteomics, the glycoproteomics analysis is mainly carried out by Li Wei Cao from Johns Hopkins, with identification of over 11,000 proteins, 51,000 phosphoside, and over 31,000 glycopeptide. To address characteristic low neoplastic cellularity of a PDAC, we use a histology-based approach by Ralph Lubin, as well as DNA methylation and RNA-based molecular deconvolution method analyzed by Dan Trezo from Washington University to estimate a neoplastic tumor cellularity. This estimation of neoplastic cellularity significantly correlated with KRAS-Wolf estimate. In particular, KRAS-Wolf was highly correlated with DNA methylation-based deconvolution. So this data allow us to select the tumor cellularity based on the KRAS-Wolf. On the top 75% tumor are used for the further data analysis. Genomic and landscape of cohort of 105 high purity tumors show the tumor muted gene are KRAS TP53, ThinK, and 2A, and SMAD4. This is consistent with the TCGA. The specific KRAS hotspot mutation distribution is shown in the pie chart, with G12D, G12R, G12V being the most common mutations. None of the high purity tumor purity samples are G12C mutation, which is the only druggable KRAS mutation. A mutant impact with RNA and the protein level show that TP53 and the SMAD4 have the most trans effect to other genes, with a severe effect only captured at the protein level. There is a strong RNA and the protein cis effect for RBM10. When look at the cis and trans effect at the phosphorylation level, we notice that observation that SMAD4 and TP53 dominate this effect. In this case, most of the trans effects result in a higher phosphorylation level. Interestingly, mutation of TP53 seem to lead to higher phosphorylation of protein involved in DNA damage repair. Protein genomic subtype of 105 high purity tumors using unsupervised multi-omics subtyping of a gene copy number, mRNA, protein expression, phosphoside and glycoside abundance, cluster tumor to two subtypes. The two clusters show significant overlap with RNA subtype with significant choromorphic basal-like and classical subtypes. Only 11 tumors are switched from basal-like and classical subtype. PDAC is deadly cancer, with 80% of patients are diagnosed in late stage. The identification of reliable molecular features for early detection is necessary. Global proteomics level identified 21 proteins with increase in abundance. Glycoproteomics analysis of PDAC identified 75 glycoproteins upregulated in tumor. Of this, 57 were previously reported in the pancreatic cancer database, and 11 were newly identified in this study. 48 out of 75 tumor-associated glycoproteins were further validated by data-independent acquisition included in this study. Mucin type only glycoproteins also show association with the PDAC. Protein phosphorylation involved in various cellular processes, which are regulated by kinases and the phosphatases on the respective phosphosubstrate. To discover the phosphorylation event that were relevant to PDAC as well as a potential therapeutic target, we utilize phosphosite abundance to examine the relationship between phosphosubstrate and their associated kinases. BAT serum 134 was identified as one of the highly ranked phosphorylation events in most PDAC tumor. A pulse induced by BAT is inhibited upon phosphorylation of BAT serum 134 by PAC1, thus promoting cell proliferation and surviving. PAC1 was identified as one of the highly elevated kinases in PDAC in this study. PAC2, another member of the class 1 PACs, also show upregulation. PAC2 was responsible for the elevated phosphorylation of MAP kinase 6 serum 189. The phosphorylation processes is critical for MAP kinase 6 signaling. Given the importance of the class 1 PACs to PDAC, combined the inhibition of PAC1, 2, and KRAS downstream pathways will likely increase therapeutic effect. Analysis of glycoprotein expressed based on different KRAS Hall mutations identify that CEA CAM5 and CEA CAM6 were significantly upregulated in tumors with KRAS G12D, G12V, and Q61 mutation, but not in G12R mutations. The identified glycopeptide changes could be impacted by both glycoproteins and glycans that are modified by glycosylation enzymes. The alteration of glycopeptide mainly positively correlated to the global protein expression of the corresponding glycoproteins. However, heterogeneity of glycopeptide abundance was observed from the same protein due to the different glycan modification. Overall, glycoproteins upregulated in tumor were mainly modified by complex glycans with salicylic acid and or fucosylated glycans. Downregulated glycoprotein tumor were mainly modified by oligomannose. Compare the glycosylation enzyme expression reveal upregulation of several glycosylation transferases, including ST6 GAL1X3, ST3 GAL1 for3 for 11 before GALT1 before GALT2 and ST6 GAL1. Some of the glycosylation changes are only observed in protein level, not in transcriptomics level, highlighting the added value of proteomics and glycoproteomics. ST6 GAL1 and ST3 GAL1 are responsible for protein cellulation, while FUT3 and FUT11 are responsible for protein fucosylation. In line with our observation that PDAC upregulated proteins are mainly modified by silylated and or fucosylated. Inhibition of this enzyme will likely attenuate and increase the cellulation and fucosylation of glycans that were found on most of the PDAC proteins. To dissect PDAC microenvironment in the context of immune infiltration for immunotherapy, we clustered the cohort of all 140 PDAC tumors based on tumor microenvironment. The sample were clustered into 4 clusters. Expectedly, the majority of the tumors are immune cold, except that a small number of tumors in cluster D had relative high immune content. Immune hot samples were also enriched with endothelial cells. Endothelial cells are blood vessel component and needed for immune cell infiltration. We find that immune cold samples have reduced endothelial adhesion proteins and upregulated VEGF and hypoxia pathway. Both RNA and the protein analysis show that the immune cold tumors had a higher level of glycolysis and Warburg effect. Based on this data, we propose a working model where the upregulated proteins by upregulated VEGF and the hypoxia pathways are associated with depletion of normal endothelial cells, increase glycolysis Warburg effect, and a possible increase in cell junction. This event collectively inhibit immune infiltration and immune function. Targeting endothelial cells reverse and glycolysis might be an effective therapeutic strategy to improve the immune cell environment for PDAC. So in summary, we use an orthogonal approach to determine the neoplastic cellularity. Protein genomic characterization review the functional impact of genomic alteration and multi-omic subtypes. Photoproteomics uncover putative therapeutic target downstream of KRAS. Multi-omics integration link endothelial cells remodeling and glycolysis to immune exclusion. In this study, we also identified proteins and the glycoproteins as a candidate for early detection or therapeutic intervention. All the data, the raw data processing, as well as software used in this study are included in the publication with the link place for downloading. So I would like to end my talk by acknowledging people who've done the work from Johns Hopkins and our collaboration, close collaboration with Baylor College of Medicine, Washington University, Broad Institute. This project is supported by CPTAC consortium and funded by NCI CPTAC. Thank you for your attention.
Thank you. Our next speaker is Dr. David Ding. He's Associate Clinical Director for Innovation and Associate Professor of Medicine in Mass General Cancer Center and Harvard University. Thank you.
Thank you so much, everyone, for having me here, and thank you to the organizers. So today I'm going to be talking about pancreas cancer plasticity and heterogeneity, and what we've been working on at the MGH. Here are my disclosures. I do have to disclose that I'm a founder of the CTC company called TelBio, but none of this work was supported by by that company. So epithelial mesenchymal plasticity, it drives tumor cell heterogeneity. We sort of have known about this since the times of learning about EMT from the greats like Bob Weinberg. And so, you know, I think most of us know that this transitional state is in between the two. And when we do single cell RNA-seq across multiple studies, including our lab and many others, we see this as heterogeneity. And so really heterogeneity is really a reflection of the fact that cells are going back and forth in between these two states. And this is something that we've seen over many of the transcriptomic and genomic analyses, starting all the way back from Eric Carlson in Nature Medicine, showing this epithelial mesenchymal subtypes that we sort of dichotomize in clinical medicine in some of the studies that we do. But in reality, this is truly, you know, not a single cancer. And so here's some evidence where we did RNA in situ hybridization. So this is visualization of epithelial genes shown here in red and mesenchymal genes shown here in blue. And here are two xenografts that were derived from patient-derived cell lines. And you can see the first one, PDAC 6, the more red one is very epithelial. But the mesenchymal cell line or the quasi-mesenchymal cell line, which I think is more appropriate, you see both blue and red cells. So we know that these cells are interconverting between these two states and then from a clonal population in cancer. And this is not just for pancreas cancer. This is showing the original ISH was developed in breast cancer in a paper that was published 10 years ago now. And so with that knowledge, one of the areas that I had worked on early on, and I'm just going to summarize here, is circulating tumor cells. So the cells that are metastasizing through the bloodstream. We had done this in the sort of KPC model, the P3 flux model. And we had developed a way to do single cell RNA sequencing on these samples. And from this study, what we found was that EMT was enriched in these circulating tumor cells. But when we dug deeper into those cells, we actually found that those cells were stuck in between this E and M state. So they weren't really epithelial, they weren't really mesenchymal, they were somewhere in between. So this biphenotypic state is something that we saw in vivo in these mouse models as well as in patients. Now, when we stained for those same markers in the primary tumor, what we found was interesting was that a lot of those CTC markers were enriched at the tumor stromal interface. And this set off a line of investigation in the lab of studying, well, is this where CTCs are born? And that led to this experiment that we used patient-derived cell lines, made them GFP positive. We made fibroblasts that we turn immortalized and made M-cherry positive. And we asked the question, does this induce single cell heterogeneity? And in this work, which I'm just going to summarize again, since many people have known about this particular work, what we found through single cell RNA sequencing and CyTOF was that these single cell populations exist on this state of E to M transition. In this particular study, we also found that we could visualize these cell states in individual glands and tumors and describe intratumoral heterogeneity of E and M states, as well as proliferation. Now, one of the things that we found that was important in this paper that is important for the rest of this talk is that we did find that TGF-beta secreted from calves, again, this is not something that I think is just unique to what we've found, many others have seen this, drives some of this PDAC EMT behavior. So there's not only just tumor intrinsic properties, but tumor extrinsic properties of the tumor that drive this EMT phenotype in cancer cells. One of the other things that we were studying at the time as part of a Stamp to Cancer team was what was the effect of FOLFIRINOX or combination chemotherapy on PDAC EMT states. In this experiment that was published by Rebecca Porter in my lab, we had taken patient-derived tumor spheres, these cell lines that were derived from patients, did RNA-seq and looked at what did FOLFIRINOX do to them. And what you could see going from red to blue with RNA-seq is that all the cell lines, irrespective if they were epithelial or quasi-mesenchymal, they became more mesenchymal in the setting of FOLFIRINOX state. This suggested that these cells were probably induced into resistance by seeking the mesenchymal state. Now, when we developed this RNA-ish, we actually developed it to be a CLIA certified clinical test. This is now completely deployed at the MGH. We stained primary tumors of patients that were treated with FOLFIRINOX in the neoadjuvant setting and resected, and compared it to untreated resected controls. And what we saw was the same thing: enrichment of mesenchymal tumor cells in the setting of neoadjuvant FOLFIRINOX, indicating that the resistant population in the setting of combination chemotherapy in preclinical and clinical samples was this EMT transition. So with that background, as part of the Stamp to Cancer sort of neoadjuvant pancreas cancer team, we completed a multi-institutional randomized neoadjuvant trial of FOLFIRINOX, FOLFIRINOX plus losartan, or FOLFIRINOX plus losartan nivolumab. And just for reference, losartan was found to be an indirect TGF-beta inhibitor that was evaluated, and this was really studied in Rakesh Jain's lab at the MGH. Nivolumab, as everyone knows, is anti-PD-1 and immunotherapy. And the goal of this was to determine if we can increase R0 resection rate in localized, advanced, unresectable pancreas cancer population. This trial is complete. I will not be talking about the clinical outcomes of this, but we are sort of encouraged by some of the things that we're seeing. What I am going to tell you about is what did we do on the science side. So this was our biospecimen correlative science plan. We took endoscopic ultrasound biopsies from patients, and our priority was organoid formation. This was sent to Hopkins in Rick Burkhart's lab, where he was successful in developing 23 organoids from 63 patients that we did biopsies on. Not all of those were actually sent due to the COVID pandemic, but we were able to do whole exome sequencing and RNA-seq on those pre-treatment biopsies. In addition, we collected over 1,500 samples of both STREC or EDTA plasma for correlative blood analysis. And we did analyze resection specimens across arms, 36 patients with EMT, RNA in situ, and spatial transcriptomics, which is what I'm going to talk about today. And so the first thing is that pre-treatment, treatment-naive specimen, we had fresh frozen, so we only were able to do RNA-seq and whole exome. And the bottom line here is that across those different treatment arms, we did not see a difference in baseline differences in classical or mesenchymal or basal-like subtypes. And we saw exactly the proportions that we expected. About 70% were classical epithelial, about 30% were mesenchymal. And so this really told you there was no sort of biases in our randomization for patients before treatment. But one of the things that we wanted to look at is after treatment, those specimens that we took out, what happened? And so we had developed this EMT-ish. This is the CLIA-ish that we had developed. And in this case, red is the mesenchymal markers, brown is the epithelial markers. It turns out that artificial intelligence is not intelligent enough to find tumors, so we still needed to mark where those tumor glands were. And Catherine Zhu and Evan Lang in the lab had sort of marked this in approximately 20 glands if possible per patient. We then use Halo AI to then quantify the number of cells in each of those glands and then classify, are you epithelial or mesenchymal? And this is the data. So each column here is a patient specimen at resection. Each on the y-axis is the percentage of quasi-mesenchymal cells per gland, and each dot is a gland. And you can see there's a lot of heterogeneity across individual patients in terms of these glands, in terms of their M or E states, but also that some tumors, like the ones on the far right, are more epithelial, while the ones that are on the closer to the mesenchymal side are on the left. And so when you have this type of data, you can start doing some interesting things, and this is just early days, but what we did was we said, okay, let's make each circle here or each pie chart, the size of the pie chart is the number of cells in the gland. And the relative red is the percentage that are mesenchymal, and the relative blue is the relative epithelial. And so we now are able to start looking at intratumoral heterogeneity and start asking these questions if your glands are small, are they more epithelial? Or if your glands are large, are they more mixed, or are they more mesenchymal? And so this is the type of data that we've been generating. But we wanted to ask the simple question of, compared to FOLFIRINOX arm 1, is the FOLFIRINOX plus losartan, the TGF-beta modifying drug, was there a big difference? And when you saw the percentage of EMT glands in each of the arms, we see a trend of lower mesenchymal glands in arm 2. When we use a threshold of 5% of a gland to be called mesenchymal, we saw that per patient, we would see these sort of trends being a little more stark in arm 2, which is the losartan containing arm. And so altogether, this suggested that TGF-beta inhibition may be altering EMT states in the resected specimens that were treated in a neoadjuvant randomized trial. So we decided we wanted to go deeper, and so we did spatial transcriptomics with the NanoString geomics platform. This is FFPE specimens that we then used a whole transcriptome assay of about 18,000 genes that are then laid on top of the tissue. We then use fluorescence stains to identify regions of interest that are used to then get those RNAs out for analysis. And so this is how it looks. Again, this is in collaboration with Will Huang. He has 2 great people in his lab that recently presented posters on this type of work. But what we're doing here is now staining for the cytokeratin positive cells in green, the immune cells in magenta, or the fibroblasts in teal, and then taking the whole transcriptome of each of those areas of interest and analyzing what happened. And so this is the data to date. And this is just showing unsupervised clustering of these different regions. And what you can see is that the epithelial compartment separates from the immune and the fibroblasts, just showing that our data quality is good. And this is being led by Amai Pankaj in the lab. And the first question we asked is, if we just do the Moffitt gene signatures, basal, mesenchymal, or classical epithelial, what you can see in arm 1, which is the blue area, is that they were mostly enriched for mesenchymal tumor cells, consistent with that ish that I just showed you. While the arm 2, the losartan containing arm, was mainly enriched for classical epithelial. Again, sort of supportive of that EMT ish, but now at sort of higher resolution. So this really indicated that losartan may be altering TGF beta activity and EMT status in the neoadjuvant setting of humans. This is just another way of looking at the data, but doing it in a supervised way. We're just comparing arm 1 glands to arm 2. And again, TGF beta came out as significantly different. So the FOLFIRINOX arm, TGF beta is higher than the FOLFIRINOX losartan arm. The arm 2, the losartan containing arm, the tumor cells were more enriched for OXPHOS pathways and adipogenesis. Again, something that we're looking into deeper. So the other thing that you can do is look at the immune cells, which we did. And when you do immune cell deconvolution, we saw that in arm 1, we saw increased macrophages compared to arm 2. And in arm 2, we saw increases of CD8 T cells as well as endothelial cells, indicating that there are more T cells coming in the losartan containing arms. Again, all these things seemed encouraging that we are having a biological effect of losartan in these tumors. Interestingly, when we looked at the CAFs, we didn't see many differences. This was something that we did not expect, but as they say, the data is the data. And the last thing I wanted to talk about is, future directions that we're going. We're using a single cell molecular spatial profiling technology called the NanoString Cosmics. This, unlike the prior assay, is only 1,000 genes, but in this case, you can visualize those 1,000 genes on the slide itself. So you have single cell resolution of these 1,000 genes. And so what you can start to think about is that we can now build this Google map of pancreas cancer. And so you can do this UMAP clustering of those 1,000 genes and sort of identify different cell types based on that. But then you can map them spatially back to the tumor histology or the tumor image and know where these are in space. And so this is the type of data that we're generating. Still trying to build the tools of how to use this type of data, but it's something that we're very excited about. And on top of that, we needed to figure out how do we make this data more accessible to people. So my good friend Martin Arye at the Dana-Farber, he's was at the MGH Cancer Center, but he's a computational scientist. And he basically took the Google Maps platform and uploaded the Cosmics data and created this viewer, and you can sort of toggle around and drive around. You can't quite walk in the tumor like you can on Google, but it's pretty good. So one of the things that we're focused on is, can you start finding patterns that are interesting for people's research? So in summary, neoadjuvant FOLFIRINOX really induces this mesenchymal PDAC. The losartan arm does look like we're having some biological effects. And we have a lot of other studies that are ongoing on this neoadjuvant trial. Many, many people to thank in my lab, some of my alumni as well have come from lab, and of course, Will Huang over there, who was instrumental in the spatial work. Thank you very much.
Thank you very much, Dr. Ting, for the excellent talk. The next speaker is Dr. Kenneth Olive. Dr. Olive is from Columbia University. She's associate professor and also the director of the Oncology Precision Therapeutics and the Imaging Core, shared resources. His presentation is ferroptosis of PDAC, animal models of PDAC. Thank you very much.
Thanks very much for the opportunity to speak. And obviously, as you can tell from my cover slide, I've been quite rude and performed a bait and switch. And I will not actually be speaking about ferroptosis today. I apologize. Everyone can get up and leave if you want. But I would like to say that what we are going to talk about is also cancer metabolism, and it's entirely unpublished, every slide, and also actually entirely unpresented. So this is the first time it's into the wild, and hopefully you'll find it interesting. So we're going to talk about this protein BMAL2 that my lab has come to focus on, finds a little bit interesting. Maybe you will as well. So we came to this protein by asking a very top level question. What are the key drivers of pancreatic cancer malignancy? Very open-ended, squiffy sort of topic, but we came at it using a specific approach of looking at regulatory network analysis or RNA transcription factor activity. Very quickly, we're using RNA expression data in a different way. Instead of just looking at what genes are expressed in different groups of samples and performing differential expression analysis and then looking for patterns like you stare at tea leaves in your cup, we are using an approach to actually take RNA data and use that to infer the activity of transcription factors, right? Because think about it, if you're an enzymatic activity assay for transcription factors would need to know the abundance of products and reactants, which are all RNA transcripts, which you measure using RNA-seq. So if you know what the target genes of our protein are, the positive and negative, the transactivating and trans-repressing targets of a transcription factor are, you can infer its activity. And I say RP here to generalize the term, no longer just transcription factor, but also chromatin modifiers and cofactors, things like that. And so if you have two samples and you have one where all the positive targets of a transcription factor are high and all the negative repressing targets are low, and then a second sample where it's the other way around, it doesn't take any math to look at that situation and say, yeah, the activity of that transcription factor is probably a little higher in sample 1 than in sample 2. You don't need any math, but you could wrap it up in math, provided that you knew what these arrows are, if you know the targets, positive and negative. And it turns out that my colleague, Andrea Califano, and others in the field have found ways to infer those arrows computationally, de novo from data, using large numbers of gene expression profile based on the field of information theory from mathematics. And I'm not, for lack of time, I'm not going to go into how this all works, but I can say that the approach is extremely experimentally validated. And so what that does is give us a scaffold, a list of all the transcription factor target genes in the genome, to which we can then say, all right, now we're going to convert gene expression profiles to transcription factor activity profiles. And this has all sorts of statistical benefits. I'm not going to go through it in detail, but what I will say is, as a biologist, what this means is that when we do a comparison between two groups of samples, we get built-in mechanism. And that's something that has been sorely lacking in differential gene expression analysis since its inception. Transcription factors drive phenotypes. That's what we know from biology. And so now we can perform what we call master regulator analysis, where we take profiles that represent two different phenotypes, a transition between two phenotypes. When we say, what are the transcription factors whose activity is most upregulated or downregulated as you go from phenotype A to phenotype B? And we can use that to now ask this question: what are the key determinants of PDAC malignancy? But of course, what does malignancy mean? That's just a word. We had to define phenotypes that would represent malignancy in pancreatic cancer. And so what we came up with were 4. The transition from benign precursors to adenocarcinoma, so IPMN and PANN, low grade. The transition from low grade cancer to high grade cancer. The transition between long and short survival within pancreatic cancer patients. And then, because it's mutated in 95% of cases, association with mutant KRAS activity. And in order to do this and to avoid the complexity of grinding up tissue and getting tumors that are 70% stroma, we actually performed laser capture microdissection and RNA sequencing to isolate just the malignant epithelial cells and also the stromal samples from almost 200 human pancreatic tumors, as well as 45 low-grade precursor lesions. And very gratifyingly, when we splay that out in an unbiased way by principal component analysis, principal component 1 separates between the epithelium and the stroma, and principal component 2 separates between precursor and cancer. And I'm not going to talk about the stromal samples today. I'm just going to speak about the epithelium. And so when we now look, and this is now what was principal component 2 on the prior slide, when we just look at the differences within the epithelial samples, we see that low grade, actually precursor lesions are all at this end of the principal component 1, and low grade cancer and then high grade cancer towards the right. And that correlates with solid growth patterns. It correlates with the more aggressive Moffitt class. And so this is all well and good. What about transcription factor activity for known transcription factors, things we already know are involved in the disease? Well, P53 and SMAD4, when we look at their activity along this path, along the scale, goes tremendously down, as would be expected for 2 genes that are mutated and inactivated in this disease. Same thing for transcription factors associated with GI fate, right? So you see a loss of GI identity. You see a hyperactivation of a number of transcription factors that are known to be oncogenic and to promote growth in pancreatic cancer. And same thing for genes that are known to promote the squamous identity within pancreatic cancer. All right, so the things we expected to be there are there. What can we find now by looking at these 4 phenotypes? So the transition from precursor to cancer, the transition from low grade to high grade, turns out that the genes that do one are highly correlated with the genes that do the other. So there's some parity of transcription factors that promote the progression or restrain the progression from low grade to, from between initiation and progression. Similarly, genes that are involved, that distinguish tumors with poor survival are enriched in the high grade tumors as opposed to precursors, again, as expected. And then we also have a list that are associated with RAS activity. And when we integrate those 4 lists, put them together, now we have our overall list of master regulators of pancreatic cancer malignancy. And we look at the first one on the list, and it's a protein I've never heard of, ARNTL2. And so I immediately go and Google ARNTL2, which of course I've studied for now 2 years, and they changed the gene symbol on me a month ago. So I'm trying to learn how to say BMAL2, and I might refer to them interchangeably in this presentation. It's a circadian rhythm gene. Good, we figured out pancreatic cancer, right? I'll just get better sleep and we're gonna cure the disease, right? All right, what's going on here? So this is a protein that's a very close biochemical homologue of HIF1 beta. HIF1 alpha and HIF1 beta form a heterodimer and they drive hypoxia classically. And similarly, ARNTL2 or BMAL2 forms a dimer with clock, one of the drivers of the circadian rhythm pathway. And when we look at the positive targets of BMAL2, they are most highly enriched in pancreatic ductal adenocarcinoma. And when we look at the negative targets, they're most highly enriched in precursor lesions, as expected. That was part of the screen that we put in. One quick thing we wanted to do is make sure this wasn't some sort of statistical fluke of the data set we were using. So we looked at all of these phenotypes across every data set out there and found that initiation and progression or survival associations are all correct and reproduced in multiple external data sets. And then because there's multiple classifiers that have been published, we went and looked at every classifier across every public data set and took the more aggressive versus most aggressive versus least aggressive molecular subtype in each of those and said, what about the activity of BMAL2? In all cases, it was associated with the more aggressive subtype. So we now can say that BMAL2 is reproducibly, its activity is reproducibly associated with pancreatic cancer malignancy. But what does it do? All right, so we did the typical thing, but we looked in this case at instead of gene set enrichment at tumors that have high activity versus low, what we did was just say, well, what are all the target genes that were inferred for this transcription factor and what gene functions do they associate with? And we saw association with hypoxia and with glycolysis and also with KRAS, which is again expected from the way we perform the screen. And so I had my team go and pull a hypoxia gene signature that's highly published from Celeste Simon's lab and say, are there connections between those genes and BMAL2? And it turns out that BMAL2 controls the expression of every single protein in the hypoxia gene signature by no more than 2 degrees of control. which said, all right, maybe we should think about hypoxia, but it was certainly very exciting for me because my lab has been studying drug delivery and perfusion in pancreatic cancer and vascularity for more than a decade now. And we have this old slide looking at a pancreatic tumor in our mouse model, a genetically engineered KPC mouse. And we can look at a contrast agent. Unfortunately, the video is not going to work. They were working on getting the codec for it, but that's okay. I'll just tell you, what's that? Yeah, no, it's not going to work. That's okay. It's fine. What I can tell you is the tumors are hypoperfused. And in fact, when we look at the tissue, there are fewer blood vessels within the, the frank parenchyma of pancreatic cancer. These are old data. I don't want to dwell on it. The extension of that is that one would infer that they are hypoxic, but in fact, we've never really measured that very well. So we went and bought this machine called an oxylight sensor. It's a gold standard way of measuring partial oxygen pressure in tissues. And you literally can physically insert this needle into the tumor. We do this image guided so that we're not opening the abdomen in any way. And this bright reflection is the needle going into the tumor. And we can measure multiple points within the tumor and do this across multiple animals while they're either inspiring regular air or oxygen. And you can see that the level of partial oxygen pressure in these tumors is obscenely low, less than 1 millimeter of mercury. There is no other mammalian tissue that lives in that environment. This is nearly anaerobic. I mean, nearly, ischemic levels, right? And so how are these cells living? And also, does this redefine the concept of Warburg, which is usually considered aerobic glycolysis, but we're looking at anaerobic glycolysis potentially in a setting of something that has that low oxygen. And in fact, there is one paper in the literature where some years ago, there was an attempt to make these measurements in human patients, 7 patients using a sensor that was outside its dynamic range. So no one believed the results because the data were so low, but they're completely aligned with our mouse data. And now we're fixing to do this in human patients with this kind of more gold standard technology to affirm it. So that is such low oxygen pressure that you cannot achieve it in a hypoxia chamber. They leak too much. You can't do it. So we had to develop, use these newer chambers where they're closed systems and pump in obscenely low levels of oxygen. So, okay, maybe BMAL2 is involved in hypoxia. We know that it's a close homologue of HIF1 beta. We know that it can potentially heterodimerize with HIF1 alpha through old data in the literature, old biochemical data. BMAL2 has numerous hypoxia and glycolysis targets, and the disease itself is extremely hypoxic. Is it doing something? So when we knock it down or knock it out, we see a massive change in glycolysis gene signatures. In fact, it's the #1 signature that we see on our list. And so we went ahead and made knockouts and looked at their viability and their migration under hypoxia. Both of them are deficient, but this was the killer for me. This is just a glycolysis assay looking at lactate secretion in the knockout cells versus wild type cells under normoxia in black versus hypoxia in red. And normally when you go from normoxia to hypoxia, you see an increase in glycolysis and increase in, that's a section, secretion. When we do this in the context of the BMAL2 knockout, you see a massive defect in glycolysis and you see a massive loss of lactate dehydrogenase, the key regulatory enzyme of glycolysis. So what's going on here? Oh, I should mention that GAPDH, which we normally use as a loading control, is a direct target gene of BMAL2, and so it's dysregulated as well. What's going on here? So this is a beautiful slide from Alec Kimmelman, who kindly donated these data to the story, where he did a genome-wide CRISPR screen for genes in the genome that are needed for viability under hypoxia. And he found that that same protein, LDHA, is the most important for hypoxia. Unfortunately, BMAL2 was not in his library, so we don't have the data for that one gene. SLC2A1 or GLUT1 was the second on his list, and it is a direct target of BMAL2. And so we started thinking, well, if this is the canonical setup, BMAL2 is thought to bind a clock, HIF2 beta to HIF2 alpha, HIF1 beta to HIF1 alpha, but there are data in the literature suggesting that this is possible. Maybe BMAL2 does this job in pancreatic cancer. So let's look at HIF-1 alpha. And this is the killer experiment. Normally under hypoxia, HIF-1 alpha is induced, but in the context of BMAL2, complete loss of stabilization of HIF-1 alpha quite potently, whereas HIF-2 alpha actually goes up a little. And HIF-1 alpha and HIF-2 alpha are not perfect homologs. They actually have vastly different effects on hypoxic metabolism. They control totally different programs. So it is possible that this is forming a switch between these two different modes of hypoxic metabolism. And that's the thing that we're chasing up right now. I will say that when you think, returning to this idea that the pancreatic tumors may be existing in this extreme state of hypoxia and hypoxic metabolism, that they may be even more dependent on lactate dehydrogenase than other tumors in vivo under this extreme hypoxia. And indeed, when you treat, and again, these are data by Al Kimmelman, not my lab, when you treat with a lactate dehydrogenase inhibitor, multiple different human pancreatic cancer cell lines are selectively sensitive under the context of hypoxia. And so we're continuing to chase this up, but with that, I'm happy to take any questions. I do want to highlight the contributions of Carlo Maurer, who developed the whole laser capture data set and master regulator analysis in our lab, and Alvaro, who's been working on the actual experimental side of this story. That, happy to take questions.
We will hold the question to the end.
Oh yeah, I guess we're doing questions at the end.
Yeah. Thank you, Ken, for the wonderful talk. And our last speaker is Castor Lysacier from University of Michigan. He will join us on Zoom. His talk, title of his talk is Metabolism of a PDAC.
Hi, everyone. Really so bummed that I can't make it with you all in person. Busted my knee playing soccer and I can't travel. But to keep this entertaining and the audience engaged, Dr. Ken Olive has generously agreed to provide an interpretive dance rendition of this presentation in parallel. Let's encourage Ken to the front of the room with a warm round of applause. A kid. Seriously, since I can't make it, Min suggested that I provide this pre-recorded talk virtually. Thanks, Min and the organizers, for the opportunity to highlight new work from our group, as well as to highlight the trainees who led these studies. Today, I will be discussing metabolic networks in the pancreatic tumor microenvironment. Pancreatic tumors consist of an array of cell types. They are notoriously hypovascular. As a consequence, nutrient access is deregulated and the tumors adapt their metabolism accordingly. A major focus of our lab is on the non-cell autonomous metabolic adaptations that facilitate tumor growth and survival. For simplicity, we group these interactions into three flavors: cooperative, competitive, and those that mediate therapeutic resistance. These mechanisms are perhaps best described by way of example, and I will use this opportunity to highlight work from our group. Cooperative interactions are those where one cell type provides nutrient support to a second, like the sharing of amino acids or even extracellular matrix that can be metabolized, including the polymer hyaluronic acid. Competitive interactions are largely used to restrain the activity of the anti-tumor immune system. Like cancer cells, cytotoxic T cells also require nutrients to proliferate and mount effective responses. We have found that a major mechanism by which immune suppression is mediated is through amino acid degradation. And lastly, are metabolic mechanisms that mediate resistance to therapies. These include immunotherapies and chemotherapies, as we have described for gemcitabine and mitochondrial poisoning. The story I'll share today concerns uridine metabolism, or uridinolysis. This work was co-led by Matt, Zaribe, Pete, and Pawan in the Sadnan lab. I will also point out that Zaribe is on the job market, so departments with openings, please reach out. I'd love to connect you with this rising star. In short, the authors demonstrated that uridine is present in the tumor microenvironment and serves as a novel metabolic fuel to support tumor growth under glucose-limiting conditions. This project started with the intention of defining the full spectrum of nutrients that pancreatic cancer cells use. To this end, we employed the Biolog nutrient profiling assay and screened 175 lyophilized arrayed metabolites in 22 pancreatic cancer cell lines in nutrient-restricted media and assessed metabolism read out as cellular NADH reducing potential. This led to a seriously enormous and rich data set that we then interrogated for novel interactions. From this list, we prioritized by correlating individual metabolite profiles with metabolic enzyme expression to define new metabolite enzyme connections. I summarized that briefly here in this one slide. For more on this screening strategy, I encourage you to check out our preprint. Our lead gene metabolite pair was uridine phosphorylase and uridine as pictured. We observed a correlation between relative metabolic activity of uridine and the expression of UPP1. This correlation was not observed for the other nucleosides in the screening deck. Interrogation of publicly available datasets revealed that UPP1 expression predicted for worse outcomes in pancreatic cancer cohorts. UPP1 expression was also correlated with mutant KRAS expression in the TGCA. And mining of a data set that we previously deposited using the iKRAS mouse illustrated that activation of mutant KRAS in murine tumors led to enhanced UPP1 expression. Collectively, these data encouraged us to take a deeper look at uridine and UPP1. From the screen, our data revealed that uridine could fuel metabolism in glucose-limited media. In fact, this concept had been reported previously, as far back as the 1980s, where the thought had been that uridine supports metabolism under glucose-limiting conditions by promoting nucleotide synthesis. While our data with UPP1 support this concept, uridine is actively catabolized in the media and inside of cells into uracil, releasing ribose. Our screen was designed to identify metabolites that supplied the cell with reducing potential, here as NADH, which is beneath what was previously considered in nucleotide metabolism and could not be accounted for by this classic mechanism. Rather, we put forth that uridine-derived ribose is used to fuel glycolysis and central carbon metabolism more generally by rerouting ribose through the non-oxidative pentose phosphate pathway. Indeed, we observe in multiple cell lines, one presented here, that uridine and glucose carbon fuel the same pathway, that this can be recapitulated with ribose, and this operates independently of glutamine. This was further validated using isotope tracing and metabolomics. Here you see we labeled the ribose carbon. In the interest of time, I will not walk through each of these graphs, but for the metabolism aficionados in the audience, I will point you toward this preprint for further detail. Furthermore, we saw near identical patterns of uridine metabolism in vivo to those we observed in vitro when we delivered this isotopically labeled uridine to mice bearing either orthotopic or subcutaneous pancreatic allografts. These results demonstrate that uridine is a metabolic fuel and it can be utilized in pancreatic tumors in vivo. Next, we sought to further investigate the role of UPP1. To this end, we knocked UPP1 out of several pancreatic cancer cell lines and observed that they were no longer able to metabolize uridine, as read out by both NADH here on the left, and the inability to convert uridine into uracil shown on the right. This was again substantiated by using isotope tracing and metabolomics, where here you see the loss of color in the knockouts. This reflects a lack of uridine metabolism. But again, in the interest of time, and for those who would like to dig into this data, we encourage you to check out the preprint. Next, we looked into the regulation of this uridine UPP1 axis. As a postdoc, Hao Chung Alec and I found that mutant KRAS drives anabolic glucose metabolism through the MAP kinase pathway. Similarly, we found that KRAS-mediated MAPK signaling also drives UPP1 expression in both mouse and human models. Furthermore, we also demonstrate that nutrient restriction augments the MAPK-mediated regulation of UPP1, which we believe makes mechanistic sense, as when cells are starved of glucose, they turn on UPP1 higher to salvage uridine to continue to support bioenergetics. As with all of the experiments, we validated this aspect in multiple cell lines. We then tested the growth of our syngeneic UPP1 knockout KPC cell lines in orthotopic tumor models. Using multiple lines and multiple sgRNAs, we observed a marked reduction in tumor growth, which paralleled a decrease in the ability of these tumors to metabolize uridine into uracil. The decreased growth in vivo was also remarkable, particularly when compared to the in vitro growth of the UPP1 knockout cell lines, which do not exhibit proliferative defects. These results suggest that UPP1 and uridine metabolism are uniquely required within the metabolic constraints imposed by the tumor microenvironment. In summary, we found that uridine is a novel fuel in pancreatic cancer, whose metabolism is mediated by UPP1. UPP1 expression is regulated by KRAS MAPK signaling, and this is augmented by nutrient availability. Finally, we show that UPP1 inhibition is growth inhibitory in immunocompetent orthotopic pancreatic tumor models. With that, please allow me to thank my awesome research team, those who led this work, a truly fantastic network of collaborators. Those who participated in this study are bolded, and our funding source. I'd also like to extend my sincere thanks for those who paid attention to this virtual talk, as well as extending my most sincere apologies for not being able to join in person. Again, I look forward to addressing your questions during the Q&A session.
Thank you very much. Now, I would like to invite all the speakers to the podium for the question and answer section. Please go to the microphone if you have any questions. I want to make an announcement is that I have been texting back and forth with our support team. Unfortunately, we cannot bring Dr. Zhang online, so she will not be here to answer any of your questions. If you have questions regarding her study, please email her after the conference. But we have all the other 4 speakers, here to answer any questions you have. Go ahead.
Wonderful talks in this session. I have 2 questions for Dr. Ting. In one of your slides, you showed that you label calves with mCherry and to look for the single cell heterogeneity. So my question is, did you choose any particular population of calf to do that? Because upcoming studies showing that calf population is so heterogeneous and of different origin, and probably all cells are invasive or non-invasive. So
Yeah, no, that's a great question. So the calf cell line is when we did single cell RNA sequencing is heterogeneous, as all the calf cell lines are. I'd say it was about 60 to 70% MyCalf and 30 to 40% iCalf based on single cell profiling. The interesting thing, which I think is in the supplement of the paper, or may not be, but with culture with the cancer cells, when we picked those calves, we did that experiment. We just didn't talk about it in the paper. They become more iCalf-like. So in the setting of culture with cancer cells, it appears that something in the cancer cells is making them more iCalf-like.
my second question is, TGF-beta is a short distance cytokine, right? In our studies, we never see a direct effect of TGF-beta on our immune cells. And you showed very direct effect. Did you see any other cytokine which was induced by TGF-beta to initiate this immune response in your studies?
Yeah, no, that's a great question. We, again, we didn't see many differences in the fibroblasts. Obviously the tumor cells, there's a lot of differences, but where's the cytokine coming from would be question number 1. The immune differences we see are sort of the cell populations are different, like CD8 T cells are higher in the losartan containing arm, but they're CD8 naive, which means that they still cannot see the tumor because they haven't generated memory, right? So, so I think those are all things that we are still left to explore. Unfortunately, we have data that we can dig through, but it's, you know, it's one of those things of, you know, even if we see at the RNA level, is it really being secreted at the protein level? So, but great
Thank you.
question.
Yeah.
So in the interest of time, we will have one question per person. Okay, because there's no break between this and the next section. Thanks.
Thank you for the great talks, everybody. It was very enjoyable. My question is for Dr. Olive. I was really fascinated by the data showing how extremely hypoxic the pancreatic cancer cells, the environment is. I was wondering if you actually looked into the possibility of the reverse Warburg effect happening. So where the cancer cells, because they're so hypoxic, are producing lactate, which is then acting on the calves to feed nutrients into the cancer cells?
Yeah, we don't have data on that yet, but actually literally tomorrow, we are shipping samples to that guy right there on the screen, looming over my shoulder, that are going to be a complete set of metabolomic analyses. And so we should have a lot of more kind of granularity about what's actually happening into the metabolism of these cells very shortly.
Sounds interesting. Thank you.
I have
Yeah.
a question for Kenneth again. This is regarding the differential regulation of HIF1 alpha, HIF1 B, HIF1 alpha, Y, HIF2 alpha, especially when your BMAL is regulating one over the other. But both these proteins are proteolytically, proteasomally degraded by the same pathway by PHT enzymes.
Yeah.
How do you explain that HIF2 alpha goes up, not HIF1 alpha?
Yeah, so there's pretty good data about, to use as an analogy, the role of HIF1 beta and protein stability for HIF1 alpha. So in addition to the E3 ubiquitin ligase and degradation, once HIF1 alpha is stabilized, it stays stabilized by binding to HIF1 beta. And so if we use that as an analogy, it may be that BMAL2 in the context of pancreatic ductal adenocarcinoma plays that role for HIF1 alpha and then perhaps not for HIF2 alpha. These are things that we're working on right now by co-immunoprecipitation to really to demonstrate. You could imagine that the upregulation might just be a simple compensatory process.
All right, we'll discuss. Thank you.
I have the question for the Dr. Choi. So you already identify MSI significantly impact the pancreatic cancer metastasis. So I'm wondering whether MSI also influence pancreatic cancer sensitivity to immune surveillance and destruction, because the tumor metastasis is always related with immune responses, right?
Yes, thank you for the question. We're working on this. I didn't have time to share it today, but we've started looking into reactive methionine changes in the immune components in the tumor microenvironment. And we're currently working on characterizing those changes. But to answer your question, yes, MSR activity changes the activation status of various components in the immune system. And we think that does have an impact in how pancreatic cancer cells are seen or not seen in the disease setting.
Thank you.
My name is Huang from MD Anderson Cancer Center. I have a question for Dr. Olive. You show the BMAL2 is hypoxia. The activity is induced by the hypoxia. Whether that activity also controlled by the RAS activity.
Yeah.
Since the RAS, we know the RAS mutation is not play much role during the mouse lifelong. And once the RAS activity go up, and then the RAS activity more play role for the tumorigenesis. So my question is, did you see the hypoxia, the RAS activity change leading to BMAL2 activity high? Whether BMAL2 expression level, you also see difference in the pan-enriching all the way to the PDAC. Thank you.
Yeah, I'm not sure I caught the last part of your question. Let me answer the first part of it, and then you can tell me if I'm missing a component. So you first asked about the the relationship between BMAL2 activity and RAS activity. And I had that slide in my talk and it was too long, so I took it out. But the answer is that when we treat our cells with any of about 8 different MEK inhibitors or RAS-MAPK pathway inhibitors, then you see a massive downregulation in the activity of BMAL2. You do not see the same with PI3 kinase inhibitors. So it does seem to be downstream of the MEK effector arm, or the RAF-MEK effector arm. And what was the second part?
The expression level during the tumorigenesis.
The pain progression.
Yeah.
BMAL2 expression level.
Yeah, I mean, it does look to be hyperactivated as you go through the progression, but we really only have the beginning and end of that process, right? We have the low grade precursors and we have the cancer samples, but we don't have, say, high grade precursor samples to look at.
Okay, thank you so much.
Sure.
Okay, I think I will stop here for the question section. Let's give another big round of applause for our speakers. Thank you for the wonderful talk, all of you. And also thank all of you for attending these sections. Wish you have a wonderful trip back home. Lastly, I want to thank my co-host, Nancy. Wish everybody have a wonderful trip back home. Thank you.
Thank you.